Here is my interpretation of the case study variant. Results of a lens abnormality panel provided us with this clinical variant to analyze and interpret. It is a heterozygous variant in the FBN1 gene. This is a coding sequences mutation where there has been an alteration in the nucleotide sequence, a substitution of guanine to adenine at position 356. The result is a novel missense mutation where there is a substitution of tyrosine by cysteine at codon 119. Physical observations of the patient included bilateral lens subluxation, tall stature, and long toes. This was a pediatric patient presenting in the clinic with these characteristics at 4 years of age. The child is male. The FBN1 gene is a protein-coding gene located on chromosome 15. It is comprised of 66 exons and is fairly large. The gene is expressed in multiple tissues, primarily in the placenta, fat, gallbladder, and ovaries. The gene provides instructions for producing a protein called fibrillin-1, as well as another protein called asparagine. Fibrillin-1 is a large extracellular glycoprotein which functions by providing structure to calcium-binding microfibrils, which aid in offering structural support in elastic and non-elastic connective tissues in the human body. These microfibrils contain a protein called transforming growth factor beta, involved in controlling growth and division of cells, key aspects of cellular development. Therefore, a mutation in the FBN1 gene, which may disrupt the formation of these microfibrils, is likely to cause some dysregulation of these processes. Crucially, the microfibrils provide a structural support of various load-bearing connective tissues, such as in the lungs, blood vessels, skin, and in the eye. In addition, fibrillin-1 regulates osteoclast and osteoblast maturation and differentiation. The other protein, asparagine, is produced by tissues that stimulate the liver to release glucose into the bloodstream. Dysfunction of asparagine has been shown to play a novel role in metabolic diseases such as diabetes and cardiovascular disease. A mutation would likely impact on carbohydrate and lipid metabolism. Omin identifies the following phenotypes associated with FBN1 mutations. For this case, we can rule out the following because they're associated with short stature. A key feature here is ectopia lentis. This may describe a key characteristic that the patient presents with as lens subluxation, a condition by which zonular fibers which hold the lens of the eye in position has been displaced, though it remains in the pupillary aperture. Under normal circumstances, the zonular fibers hold the equator of the lens and its surfaces to the ciliary body and ciliary section of the retina. The characteristics of tall stature and lens subluxation are consistent with marfanoid features. However, this is not a definitive diagnosis. A clinician may decide to look at plasma homocysteine concentration for homocystinuria, as the clinical findings presented can be very similar. In Marfan syndrome, there are two primary features commonly associated with the disease: some degree of ectopia lentis and blood vessel defects relating to cardiac function. A diagnosis of Marfan syndrome is confirmed via Ghent nosology, a set of defined clinical data. In 2010, the criteria was updated to place more emphasis on the cardiovascular manifestations. Criteria in the absence of family history includes the following. We would need to perform tests to confirm or rule out aortic root dilatation. I used Human Phenotype Ontology's Phenomizer to standardize clinical findings, and the results of all these together came out with the following, of which the two features that aligned with the FBN1 gene were ectopia lentis and a form of Marfan syndrome. The phenotypic presentation is consistent with the form of Marfan syndrome. For the review, I used ClinVar, Mastermind, and LitVar. Some citations were found, but none specific to the variant. There is still the option of looking at neighboring variants and using databases which can help to predict pathogenicity. The variant could be in the 25% that occur as a result of a de novo mutation. Ectopia lentis and Marfan syndrome both have autosomal dominant transmission, which means that the parents either possess the variant or are normal. One of two things may be likely. A novel variant may have occurred during the germline development, or one of the parents possesses the variant but is not showing full phenotypic features of the disorder. To find further information, I used Google Scholar, implementing Boolean search parameters, and I searched for the following. The search came back with 4 results. Notable findings included an example of a de novo mutation in the gene and evidence for occasional craniostenosis as an additional phenotype in neighboring FBN1 variants. Varsum uses data from UniProt to identify specific regions of the gene which have been altered by the missense mutation. There is this N-terminal domain, which normally has high specificity for fibrillin 2. There will likely be effects on interaction with extracellular matrix proteins and the TGF beta binding proteins. This can impact on the genesis, structure, and performance of microfibrils and elastic fibers in a number of tissues. The next region affected shows disruption of the interaction with MFAP4, an extracellular matrix protein produced by vascular smooth muscle cells, and this is thought to contribute to function and structure in elastic fibers in the heart and lung. It is often used as a biomarker for predicting aortic dissection in Marfan syndrome. Next, we have altered calcium binding of EGF-like 2, which have been implicated in the pathogenesis of Marfan syndrome, along with other EGF-like domains. And finally, we would expect to see an effect on this disulfide bond domain because cysteine substitutions have been shown to be frequent causes of Marfan syndrome. In silico analysis for pathogenicity prediction yielded the following results for the variant. We can assess degree of pathogenicity using the pictured population data criteria. One of the measures we use is minor allele frequency to provide information allowing us to differentiate between common and rare variants in the population. This isn't an exhaustive summary of all classifications, but where it says B, these are benign, and Ps are stronger evidence for pathogenicity. Here we have a sliding scale of strength and type of evidence. For this variant, we have identified the following classifications from Nomad. It is important to note here that the Ps are pathogenic. So here we have PM1. The variant is located in a mutational hotspot and or established functional domain. PM2, it is absent from controls. PM5 identifies an alternative variant, identified at chromosome 15 as well, and this is classified as likely pathogenic. Additionally, we have supportive evidence of pathogenicity in the form of PP2, showing the variant has a low rate of benign missense variation, and PP3, where we see multiple lines of computational evidence to support a deleterious effect. The change is fairly conserved. My final interpretation of these results is that the variant is pathogenic and damaging. It was initially difficult to assess the impact on cardiovascular functioning, but the data on interaction with MFAP4 helped to provide more evidence. Therefore, the patient would likely require prophylactic treatment with beta blockers and angiotensin II receptor blockers, as well as potential surgery somewhere down the line. Beta blocker treatment could reduce TGF activation, which is an added benefit for this condition. Before treatment, I would follow up with an assessment of cardiac function in light of the expected clinical manifestations consistent with the condition and my findings. And here are my references.